All right, we're going to pick up with part 3 of chapter 43. So, continuing on with our adaptive immune system. So, we were talking about B and T cells and how they are going through the process of development. So, we talked about self tolerance, how we want to make sure that our immune cells are only recognizing potentially pathogenic foreign invaders and not our self cells. For activation, once a B or a T cell is activated, and that's going to happen because it has encountered an antigen presenting cell, the B or T cell will undergo multiple cell divisions, so mitosis, and this process is called clonal selection, and it produces a bunch of identical cells. So they're all going to be cells that will be targeting and recognizing that one specific epitope on the antigen. When this clonal selection process happens, some of those cells will become effector cells, and these effector cells are the ones that will go on to secrete antibodies. So they are also called plasma cells. So these are B cells that have been activated. They become effector cells and secrete antibodies into the blood supply and also into the lymphatic system, into the lymph. The other subset of the B cells will become memory cells. And the memory cells are really important because they stay around for long periods of time in the blood supply, in the lymph nodes, and lymphatic system. And those memory cells are essential for mounting an immune response to the second time that this particular antigen is encountered. So the more times that you are either infected with or exposed to an antigen, specifically that one epitope, the faster and more robust your immune response will be. So when we look at this, you can see that these are B cells at the top that have different antigen receptors. So they are targeting different epitopes. So you can see this is a specific epitope that's targeted here. Here's a different B cell and another B cell. Again, these would be memory cells that would be moving around inside of the blood supply or the lymphatic system, those lymph nodes. You can see in this case that this particular B cell, this memory cell, encounters the antigen to which it is specific. So again, that specific epitope, that pointy portion that we saw before, it's going to cause clonal expansion of these cells to form either our plasma cells, which are the effector cells. These plasma cells will secrete the antibodies that are specific to that particular epitope. So these are the effector cells. The other population are the memory cells. So these are the two different types of cells that will be created through this process of activation.
Humoral immunity, carried out mainly by B cells, deals with invaders in body fluids. B cells are lymphocytes that send out antibody molecules in response to certain stimuli. There are many kinds of B cells. Each kind produces and presents its own uniquely shaped antibody receptors in its plasma membrane. These unique receptors only bind to antigens with specific shapes. The B cell that successfully binds to an antigen is stimulated to proliferate into a clone of cells with the same specificity. This process of antigen-selected proliferation is known as clonal selection. B cell proliferation produces clones of plasma cells and memory cells. Memory cells are long-lived cells that can respond rapidly to future exposures to the same antigen. They account for the speed and strength of the secondary immune response that gives us long-term immunity to a disease. Plasma cells multiply rapidly. They produce and secrete antibodies in increasing amounts, peaking about 2 weeks after the activation of the original B cell. These antibodies flow through the body's fluids and tag the original foreign antigen molecules, marking them for destruction.
So those are B cells, and that was the humoral response that we're talking about. So back to this idea of memory in the immune system. This immunological memory is very important, and it helps to protect complex organisms like us against particular pathogens, and these are long-term protections. Some of them last. These memory cells that are created sometimes last 10 years, 20 years, 30 years. The memory cells that are produced from vaccinations versus if the person was actually infected, that could vary in the length of time as well. And we'll talk about vaccinations in a little bit as well, but this immunological memory is really important for the adaptive immune system. So the very first time that you're exposed to a specific antigen, or we could say a specific epitope, that will represent the primary immune response. This first exposure could be from a vaccination, or it could be from you getting infected with this particular pathogen, whether that be a bacterial cell, a fungus, or a virus. Once you have your primary immune response, you have lymphocytes that are going to form lots of clones that are specific to that antigen. So again, your lymphocytes will be B cells and T cells that will be activated. Then you're going to have some of these memory cells that are sticking around. And the second time that your body sees the same antigen, the same specific antigen, we now have a secondary immune response. These memory cells that have been formed from the first time create a faster and more efficient response of your adaptive immune system because of the T and B memory cells that are still floating around from the first time that you saw this particular antigen. So this is showing you, again, an example here, antibody concentrations. This is time in days here. So here you have a exposure to an antigen A. So let's say that that's a particular virus. Again, exposure, perhaps you develop symptoms, maybe you don't, but your body starts to produce antibodies to that particular antigen A. They start to ramp up, they start to come back down, and now we have our memory cells. And now we see that at this point, day 28, we have exposure again to antigen A, and now here's a new antigen B. So look at this, antibody A, because there are memory cells there from before, it ramps up really, really quickly, dramatically more antibodies, more cells, more memory cells will be created as well. And not only does it increase the numbers of the cells and the antibodies released, but also it goes much faster. So you can see again from the initial response, it took, again, so say you were exposed day 2, it takes up to about day 7 before you're even starting to make these cells. Antibody production again about a week later after you've started to produce those. In this case, in our secondary exposure, it happens much faster. So within a week, you're going to have really high levels of those activated B and T cells to combat that particular infection. Again, here for antibody B, or excuse me, the antigen B, again, we're still at a primary response here. But again, if we were to have a secondary response, then we're going to have a much quicker response time here. So primary immune response takes a little while. You have a good response. It's the secondary response really where we see a dramatic increase in the numbers of effector cells and those memory cells that will be left over to help that secondary response. And this happens the 3rd, 4th, 5th time you see antigens as well. So each response just gets bigger and more intense in terms of combating a particular virus, bacterial cell, or pathogen. So we talked about this idea that the adaptive immune response has 2 sides. It's got the humoral side and the cell-mediated side. The humoral side is the B cell side. These are the B cells that are creating effector cells or plasma cells that are releasing antibodies into the blood supply or tissues or lymph, the lymphatic system. These antibodies are going to help to neutralize or eliminate those particular toxins and pathogens in body tissues. So the humoral response is combating the pathogen or the toxins due to antibodies. And then the cell-mediated response is specifically using specialized T cells. And remember, T cells are the cells that are created in the thymus. So these T cells are the ones that are destroying host cells that have been infected by the pathogen. So we're going to look at how do we go about this process of activating T cells and also our B cells. A very important intermediate cell between both the humoral and the cell-mediated adaptive immune system is a specialized T cell called the helper T cell. So the helper T cell is great because it works to activate both the humoral adaptive immune system and the cell-mediated adaptive immune system. So it's going to help activate both sides of the adaptive response. I mentioned this term before, APC, or antigen presenting cells. These antigen presenting cells are cells that typically are phagocytic. And again, macrophages, we mentioned, are good examples of things that are antigen presenting cells. These antigen presenting cells are using the MHC molecules, major histocompatibility complex molecules, and they are of two types, class 1 and class 2. So these antigen presenting cells, remember, are breaking apart the pathogen, whether that be a virus, a fungus, or a bacteria. And it's going to present, these antigen presenting cells are going to display the particular piece of that pathogen, that antigen, on the surface of the cell. And that's going to help with the immune response. So we have, again, class 2 and class 1. Class 2 MHC molecules are going to be very important in providing the molecular signature by which the antigen presenting cells are recognized. So the class 2, again, is going to be presenting the particular epitope that needs to be recognized for the immune response to take place. The antigen receptors on the surface of the helper T cells will bind to the antigen and to that class 2 MHC molecule on the antigen presenting cell. And then cytokine signals are exchanged between the two cells. And I'll show you a picture of this as well. Once this happens, once the receptor on the helper T cell binds to the antigen, cytokines are exchanged between the two. That will activate the helper T cell. The helper T cell proliferates, means makes more of those cells. It's going to form lots and lots of clones of these helper T cells. And then these helper T cells are going to go on to activate the appropriate B cells. And remember, B cells are going to be the ones that are going to make the antibodies for us. So here's an example. So you see you have your antigen presenting cell. Again, an example of this would be macrophage. You have your pathogen that's being absorbed by the antigen presenting cell, the phagocyte, breaking down the pathogen into little pieces. In this case, the antigen fragment is going to be bound with the MHC molecule. This is a class 2 MHC molecule that's displayed by the antigen presenting cell. The antigen receptor on the helper T cell recognizes this antigen. Once this connection is made, there's an accessory protein that binds here. Again, you don't need to know the name of that, but that is a protein that comes from the helper T cell. It's going to reinforce this connection. So there's two connections made between the antigen presenting cell and the helper T cell. Cytokines, which are chemicals, are released between the two. And because they're releasing these chemicals back and forth, that activates the helper T cell. And that helper T cell is going to go on to help activate B cells and also create cytotoxic T cells. So it's going to create the humoral side of the immune system. So these B cells will go on to secrete antibodies, and it also will help to activate the cytotoxic T cells these are the cells that are going to go on to help recognize infected cells and to kill those.
Now let's look at how helper T cells aid cell-mediated immunity and humoral immunity. Imagine that bacteria have infected the body. Antigen molecules on their surfaces identify them as foreign. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface along with a self protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. The antigen-presenting cell secretes interleukin-1. Both interleukin-1 and contact with the antigen-presenting cells activate specific helper T cells to divide and grow into many cells with the same receptors. The helper T cells next activate other cells in the immune system by secreting another stimulatory protein, interleukin-2. Interleukin-2 stimulates helper T cells, cytotoxic T cells, and B cells. The helper T cells divide more rapidly and increase their stimulation of other cells. Cytotoxic T cells are stimulated to become active killer cells. B cells are stimulated to develop into plasma cells that secrete antibodies against the specific antigen. Correct. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface along with a self protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. What kind of cell now responds to the antigen-presenting cell and activates other cells to attack the invaders? Click on your choice. Correct. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface along with a self protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. What kind of cell now responds to the antigen-presenting cell and activates other cells to attack the invaders? Click on your choice. Right. The antigen-presenting cell secretes interleukin-1. Both interleukin-1 and contact with the antigen-presenting cells activate specific helper T cells to divide and grow into many cells with the same receptors. The helper T cells next activate other cells in the immune system by secreting another stimulatory protein, interleukin-2. Click on the 3 different kinds of cells that can be activated by interleukin-2. Correct. Interleukin-2 stimulates helper T cells, cytotoxic T cells, and B cells. The helper T cells divide more rapidly and increase their stimulation of other cells. Cytotoxic T cells are stimulated to become active killer cells. B cells are stimulated to develop into plasma cells that secrete antibodies against the specific antigen.
So the interleukin molecule that they're talking about, those are actually specific examples of cytokines. So those are chemicals that will help the activation process. So looking at the humoral response, again, we said that the humoral response characterized by the secretion of the antibodies, and the antibodies are secreted into the blood supply and to the lymph, which is the fluid in the lymphatic system, by those activated B cells. And of course, we know that we need to activate those B cells first for that to actually happen. So we know that the helper T cells that we saw before are very important in helping to activate our B cells. So when an antigen binds a B cell, the cell's going to take in those foreign molecules by receptor-mediated endocytosis. So again, remember, endocytosis, to take something into the cell. We know that the class II MHC protein of the B cell then presents the antigen fragment to the helper T cell, and this process is essential for B cell activation. This activated B cell is then going to give rise to thousands of identical plasma cells. Again, this is the clonal expansion process. And once that happens, these become effector cells that are going to produce and secrete those antibodies. So again, we have our antigen presenting cell, and our antigen presenting cell again is going to break down pieces of the pathogen. It needs to activate the helper T cell first, and we know that the MHC2 molecule is displaying this particular piece of the antigen, so that's the epitope. Our helper T cell is going to bind along with, again, additional protein factor. We know cytokines are going to be exchanged, and that's going to activate my helper T cell. Then the next step is my activated helper T cell is going to meet up with B cells that are also acting as antigen presenting cells. So you can see that this pathogen is being bound and brought in. This is activating this particular B cell because of this MHC1 molecule. And now this B cell is activated. So the T cell, helper T cell gets activated first, then my B cell gets activated, and now my B cell can clonally expand into more memory B cells or into those plasma cells, which will secrete those antibodies. So the essential piece is that the helper T cell has to be activated first before those B cells can go on and be activated as well. So when we're thinking about antibodies, the antibodies are important. Again, those are those Y-shaped molecules that are secreted by the B cells. They don't kill the pathogens directly. Instead, they actually mark the pathogens for either destruction or inactivation with several different methods. So some examples of things that will happen when an antibody binds to a pathogen. So again, here's an example. You can see these are the antibodies that have been secreted. This is a bacterial cell that has been bound by the antibodies. So now we need to figure out what happens next, right? So the first step is that those antibodies are now bound to the pathogen, but then how do we get rid of those pathogens? Neutralization is a process where the antibodies bind to the surface of a either bacterial cell or a virus. And in the case of a virus, this helps to prevent infection of the host cell. So we know that in most viruses, those proteins that we find on the surface of those cells are essential for getting into the host cell to actually cause infection. So if you have antibodies, here's an example. So this is your picture here. So here's your virus. If you have antibodies binding to these proteins that are on the surface, this actually will prevent this virus from being able to infect host cells. So this is a neutralization process. The other thing that antibodies can do is they can bind to toxins that are found in body fluids and prevent those from entering body cells as well. So it can actually bind up any toxins that are released by viruses or by bacterial cells. Another process that's used is using these antibodies as a way to neutralize or to target the pathogen for destruction is a process called opsonization. Opsonization is where antibodies that are released from those B cells bind to the antigens on the bacteria, and this triggers phagocytosis. So phagocytosis, remember, is the process of macrophages or other types of phagocytes that are traveling around in tissues, eating up anything that is foreign. So another name for opsonization, or a way to think about it, is that it helps make the bacteria more tasty. So these antibodies help the phagocytes to find the bacterial cells and to eat them more quickly and at greater rates. So opsonization is a process where antibodies are bound to the outside of bacterial cells, and those phagocytes are able to eat them more quickly. Antigen antibody complexes can also bind to complement proteins. Complement proteins, of which there are like 30 complement proteins, help to trigger this cascade, and this process implements a or activates the complement system. The complement system is a pretty complex pathway, with, like I said, with 30 proteins, but ultimately the whole goal is to create a MAC, a membrane attack complex, and that membrane attack complex is going to create a pore in the membrane of either that foreign bacterial cell or the foreign virus, and it will lead to lysis ultimately. This is what opsonization looks like. So remember, opsonization is to make a bacteria more tasty. So again, here's a bacterial cell, here's antibodies that are bound to it. These antibodies make it easier for the macrophage to identify this as something foreign that needs to be eaten. So again, it's opsonization is to make something more tasty. And then here's the complement system. So again, complement system, lots of proteins that are activated in a cascade, we call it. And ultimately, these cascade proteins are put together and help to create this MAC complex. And this MAC, MAC, membrane attack complex, is going to allow for lysis of the bacterial cell, of which this is here, or also lysis of the virus. But this is more often that we see this in bacterial cells. So this is the complement system that ultimately creates a membrane attack complex pore, which is what this is here. So when we know that bacterial cell is busted open or lysed, of course, it's dead at that point, which is the whole goal.
The main role of antibodies secreted by plasma cells is to tag foreign molecules or microbes for destruction. Binding of antibodies to antigens can trigger several effector mechanisms. Neutralization occurs when antibodies bind to antigens, blocking their activity and making them harmless. For example, antibodies cover the surface proteins of this virus, preventing it from hooking up with the host cell. Other antibodies may bind to toxin molecules on a bacterium. The bound antibodies attract macrophages, which dispose of the invaders by engulfing them, a process known as phagocytosis. Antibody molecules have at least two binding sites, allowing them to form a clump of microbes by binding to two antigens on two different cells. This process, called agglutination, traps microbes and makes it easy for macrophages to capture them and dispose of them by phagocytosis. Precipitation is similar to agglutination, except this process clumps together free antigen molecules dissolved in body fluids. This inactivates them and attracts macrophages, which destroy them by phagocytosis. One of the most important mechanisms of humoral immunity is the activation of complement proteins. Binding of antibodies to a foreign cell triggers complement proteins in body fluids to form a hole in the membrane of the target cell. The hole allows water and solutes to enter the cell. The foreign cell swells, bursts, and dies. Humoral immunity, mission accomplished.
So all of those are mechanisms of using antibodies to allow for death of either the virus or the bacterial cell as a pathogen. So B cells, there are, like I mentioned, I think a while ago, there are different classes of antibodies, or immunoglobulins, they're called. They are similar, but they have different heavy chain constant regions. That's what makes them different. There are, like I said, 5 of them. IgD is membrane bound, so it's not going to be floating around. The other 4 are soluble: IgA, IgE, IgG, IgM. A nice question might be, which of these is not a particular type of immunoglobulin? So these are the 5 examples of the specific types of antibodies that are produced by our B cells. All right, we're going to stop there, and one more section to go for chapter 43, which will be on the cell-mediated side of the adaptive immune system. All right, thanks.